Okay, assalamu alaikum everyone. My name is Maryam. I'm a second year medical student, and today's BAS session is going to talk about muscle physiology, which is going to include both the skeletal muscle and the smooth muscle. So this is how our session is going to be structured. First, we're going to talk about the skeletal muscle, the structure, how the contraction is going to occur, and then we'll have checkpoints where I'm going to ask you like a few questions where we about the concepts that we discussed. And then we can, if any one of you joined the meeting, we can have a break and then talk about smooth muscle physiology and then another checkpoint. So we can start now. Okay, so let's talk about the structure of a muscle first. We have something called a myofiber, which is basically the muscle itself. And then this myofiber is made out of many small myofibrils. So fibrils is like a tiny fiber, and these small circles is basically a myofibril. This is a myofibril, this is a myofibril, this is a myofibril. Now the question is, what is these myofibrils made of? These are made of small contractile units, which we call the sarcomere. A sarcomere is a functional unit of a muscle. Basically, a muscle, the job of the muscle is to contract, and what does the contraction of this muscle is the sarcomere, and this is the sarcomere. This is one sarcomere, this is one sarcomere, okay? Now, the muscle cell itself, it's like a normal cell. It has a cytoplasm, it has a cell membrane. So because it's a specialized cell, it has a special name for its organs, or organelles, sorry. So sarcolemma is a cell membrane. And within this cell membrane, we have invaginations. We call them T-tubules, which we're going to discuss later on. So basically, T-tubules are just invaginations or like indentations of this sarcolemma or this cell membrane, which allows the action potential to go through and excite the muscle cell. Okay, and then any cell has a cytoplasm, but our muscle cell has a sarcoplasm, which is just a cytoplasm of a muscle cell. And any cell will have a endoplasmic reticulum. In our case, we have a sarcoplasmic reticulum, which is basically the calcium storage. Okay, so this is another picture. We have, this is the muscle cell. This is a big muscle cell, myofibril or a muscle fascicle, small muscle fiber, and then a myofibril. Okay. Okay. Now we're going to talk about the sarcomere structure. Let's have this image first. So we have multiple sarcomeres. So a sarcomere next to a sarcomere, multiple contractile units next to each other, which allows the entire muscle fiber to contract. What is the sarcomere made of? It is made of boundaries. So this is a Z, it's called a Z line. So a Z line, and between two Z lines is one sarcomere. So if we say that we have another sarcomere here, we have another sarcomere here, between each sarcomeres is a Z line. Okay, so Z line is just a boundary. And then we have what we call thin filaments. And thin filaments is basically a complex made of actin, tropomyosin, and troponin. Okay, so it's called a thin filament, and we have it right here. It's just made of actin, troponin, and tropomyosin. We're going to discuss this later, but I'm just telling you the structures. And then we have what we call a thick filament, which is the myosin head. Okay, and the M line is the line in the middle. So if you have the sarcomere is this entire structure, when a muscle contracts, contraction basically means the muscle shortens. So between this Z line and this Z line, they will come closer to each other. Okay, and this is what happens during contraction. And we're going to discuss this further in details. Okay, again, tropomyosin and troponin, these are just these are just proteins that makes up the thin filament. Okay. Okay, again, let's dive deep into the structure of the thin filament. And we have it right here. This is a thin filament. We have actin, which is the main protein that is going to play a role in the contraction. So this is the actin, the green things. Actin is made out of 13 individual actin monomers. Okay, so one actin strand is made of 13 actin monomers. So 13 of these green circles is one actin filament, okay? But the actin filament itself, or the thin filament entirely, is made out of two strands, okay? So we have one strand here and another strand here, and each strand contains 13 actin monomers, okay? Now, what is this actin? Actin, as a protein, it has a binding site like all other proteins. And what does actin bind to? It binds to myosin to make what we call cross-bridging, and we're going to discuss it later, but just know the structure. So actin, it's made out of two filaments. Each filament is made out of 13 actin monomers, And the actin, it has a myosin binding site. Okay, another thing in the thin filaments, we have two proteins. Okay, this thin red line here is tropomyosin, and tropomyosin is also a filament that covers the myosin binding site at rest, because you don't want your muscle to contract all the time. Maybe you're relaxing, or maybe you are sleeping, or maybe you are stretching. You don't want the muscle to contract, and you don't want myosin to bind to actin. And what covers it, the myosin binding site, is this tropomyosin. Okay, so the job of tropomyosin is to bind or cover the myosin binding site on the actin filament in the resting state. Okay, and then what if we want to contract the muscle? What happens is that we want to move this tropomyosin. We're going to discuss it later. Okay, but at the resting state, the tropomyosin is covering the myosin binding site. Okay, now the next protein here is what we call the troponin. And troponin is actually not just a protein, one protein, it's a protein complex, and it's made out of 3 protein subunits, or like 3 subunits. Okay, and each subunit will bind to a different different structures. Okay, so again, troponin is a complex of protein, and it just has 3 subunits, with each subunit binding to a specific thing. So one subunit is called the TNC, which is troponin C, and C stands for calcium. Okay, so it's going to bind to calcium. TNT is going to bind to tropomyosin. Okay, so as you can see here, this is the tropomyosin, the red filament. This is the troponin, and the part that is binding to tropomyosin is called the TNT. Okay. The last the last subunit is called the CNI, and I here stands for inhibits. What does it inhibit? It inhibits the actin. So in order for something to work, it needs to be active, or it needs to be activated. In the resting state of the muscle, the TNI, or the tropomyosin inhibitory subunit, will bind to actin to inhibit it from binding to myosin. Okay, so this is the troponin protein. We have this one as troponin I. As you can see here, it's bound to actin, so it inhibits actin. Okay, TNC is these two red blobs, okay, red balls, and it binds to calcium, okay? And the TNT, it binds to tropomyosin. As you can see here, this is the light blue is the tropomyosin, and this TNT is the troponin T, which binds to tropomyosin, okay? Okay, now we are done from the structure of the thin filament. Let's talk about the thick filament, which is basically just myosin. Okay, so myosin as a protein, it consists of 6 chains. Okay, 2 heavy chains, 4 light chains. Okay, 2 plus 4 is 6, so 6 chains, 2 heavy and 4 light chains. Okay, the 2 heavy chains, they intertwine to form the tail, the head, and it's basically the architectural chains for the myosin to make it like a thick thing. That's why it's a thick filament. And the 4 light chains it's divided into 2 sub chains, if you can call them that. So 2 alkali chains, and alkali, from the name, alkali like neutralizes, stabilizes, moderates. Okay, so alkali chain, it stabilizes the myosin head. Okay, and another chain, which is also a light chain, is called the regulatory chain. What does it regulate? It regulates ATPase activity. So ATPase is an enzyme which hydrolyzes ATP into ADP and inorganic phosphate, right? So this regulatory chain will regulate the ATPase itself, okay? So it doesn't, go out of control and just hydrolyzes ATP without us needing it to, okay? So these are just structures. We're going to dive deep into the details later on, but just keep in mind that myosin is a 6-chain protein made out of 2 heavy chains, which stabilizes or forms the tail, the head of the myosin, and 4 light chains, 2 alkali chains, which stabilizes the myosin head, and a regulatory chain, or like 2 regulatory chains, which regulates the ATPase activity. Okay, now this was mentioned in your lecture in the transcriptions that Dr. Shams adds to his slides, or like PDFs. So regulatory chain will regulate ATPase, but what regulates this regulatory chain? It's calcium dependent and independent kinases. So it's just to maintain the homeostasis of this chain. Okay. Okay, so this is a picture which is um, like busy with things, but what I want you to focus on here is how does the muscle start to contract. So what is prior to actin and myosin actually joining or binding to each other. So as you know, we have like action potential and action potential waves, and then we have the action potential going to the axon terminal, which allows for neurotransmitters like acetylcholine to go out of this axon terminal and then bind to, you know, the its receptor, okay, at the neuromuscular junction. It's an entire lecture. Maybe you have a past session about it, or it's another lecture altogether, but it's not our focus now. But what I need you to know is that when the action potential comes at the neuromuscular junction, the depolarization wave will allow the opening for a calcium channel, and then this calcium channel will allow for another calcium channel from the sarcoplasmic reticulum to open the calcium inside, and this calcium will go out into the cytoplasm. Okay, let's repeat this again. So you can see here the light, or like the light purple line. This is basically what? This is the sarcolemma. And this invagination is the T tubule, which we discussed. The action potential or the depolarization wave will come here and depolarizes this entire cell. Okay? Depolarization will allow for the opening of this type of channel, which is an L-type calcium channel. We will discuss it later, which will cause conformational changes to allow for the opening of this channel, which is called a ryanodine channel on the sarcoplasmic reticulum. Okay, the sarcoplasmic reticulum, if you remember from the beginning of my explanation, is basically a calcium store. It has a lot of calcium in the inside. When this ryanodine channel, which is a calcium channel, opens, it causes the release of calcium from the inside of the sarcoplasmic reticulum to the outside into the sarcoplasm or the cytoplasm of the muscle cell. And why do we need to know this? Because the first step in the cross-bridging cycle, or the actual contraction of a muscle, is calcium. Okay? Okay. Let's. This is basically what I just explained in words. The propagation of action potential, the depolarization wave will come, and then it will depolarize the C-tubules, and then it causes the release of calcium. I'm gonna talk about a little bit more in future slides, but it's called, this type of calcium release is called calcium-induced calcium release. Okay, and then once calcium is out into the cytoplasm, it causes or like initiates the actin-myosin interaction. Okay, and then the cross-bridging cycle will occur and later slides. Okay. So this is what I'm telling you about. Here you have the cell membrane or the sarcolemma. This is the T-tubule. Action potential from an axon terminal will come, and it will cause the opening of this channel, which we call dihydropyridine channel, or like a dihydropyridine receptor, which is an L-type calcium channel. Okay, this is physically attached to the receptor channel, or a channel on the sarcoplasmic reticulum called the ryanodine receptor. So calcium depolarization will come here. It senses or the dihydropyridine channel senses this change in the voltage. Will open the calcium will open, and calcium will enter. Causes conformational changes to the ryanodine receptor. Ryanodine receptor will open and causes calcium to go out from the sarcoplasmic reticulum into the sarcoplasm or the cytoplasm. Okay, so again, what I discussed, we have a channel which is called dihydropyridine channel or a receptor. It's a voltage-gated L-type calcium channel. Okay, it senses the depolarization, and this depolarization from what? From the action potential that comes at the neuromuscular junction will cause conformational changes. The shape of this channel will change. It will open, causes few calcium ions to enter. And then this will interact with ryanodine receptor, causing another conformational change to the ryanodine receptor. And what happens? This ryanodine receptor will open, causing calcium to go out of the sarcoplasmic reticulum into the cytoplasm. Okay. Now, what happens later on? Now we have calcium in the cytoplasm. What happens next is this calcium will go and bind to troponin. And if you remember from the structure of troponin, it had a subunit which is called TNC. Okay, TNC stands for calcium. So calcium go and bind to this TNC subunit. Okay. Now, this is a note that a small type of, let's go back here. This channel is the, or like this receptor is the dihydropyridine channel. a little bit of these channels, let's say 3 number or like 3 amount of these channels can cause the opening of say 15 ryanodine receptor. Okay, so a low number of dihydropyridine channel can induce the opening of multiple ryanodine receptors. And what we call this is calcium-induced calcium release, because a little bit of calcium can enter will cause a lot of calcium to go out from here. Okay, so a little bit of calcium from outside of the cell can enter the cell and cause a lot of calcium from inside the sarcoplasmic reticulum to go out into the cytoplasm. Okay, so this is what we call the calcium-induced calcium release. Okay, basically an amplification of the release of calcium, and our goal is to have a lot of calcium in the cytoplasm to initiate the contraction of the muscle. Okay, this is what I also discussed. This is the cell membrane or the sarcolemma, and we have a lot of calcium outside of the cell. Okay, so this is like 1.8 millimolar. And inside the sarcoplasmic reticulum, we have also a lot of calcium inside the sarcoplasmic reticulum. But in the cytoplasm itself, we have very low calcium concentration. So calcium from outside the cell will enter the cell or the cytoplasm, and calcium from the sarcoplasmic reticulum will go out of the sarcoplasmic reticulum into the cytoplasm. Why? Because, down its concentration gradient through facilitated diffusion through ryanodine receptors, okay, in the sarcoplasm, okay? Okay, now we reached our goal, which is having high concentration of calcium in the cytoplasm. Now what? Now what is that we will start our cross-bridging cycle or the contraction of it itself. Basically, what we have, or like what we want, is actin to bind to myosin and myosin to bind to actin. Okay, what does this, or like what helps or aids in this process, is a lot of factors, including calcium, which we discussed, troponin, tropomyosin, and also ACP. Okay, so we're going to spend a lot of time explaining this, but inshallah it's going to be easy. Okay, now at the resting state, beginning before everything, we have actin, or at the thin filament, with actin protein, with tropomyosin covering the myosin binding site, with the troponin complex, which is TNC, TNT, and TNI. Okay, and I inhibiting the actin. On the myosin head, we have the myosin or the thick filament at its low energy state. What does that mean? Low energy means it's not moving, it is not binding to anything, it is not active. But what seems counterintuitive in this case is that we have an ATP molecule binding to the myosin. And yes, it is bound to the myosin, the ATP is bound to myosin, but it's not releasing energy. Okay, so that's why we call it a low energy state. Even though we have ATP bound to the myosin head, we have the ATP is just a molecule sitting there. Okay, pretend it's like a ball or something. Okay, it's just sitting there. It's not doing anything. It's not releasing energy. So the myosin is not active. Okay, so this is in the resting state. Now, what happens is that we have calcium now. We have high calcium from outside the cell, and calcium from the sarcoplasmic reticulum all went into the cytoplasm. Now, calcium is the initiator of this cross-bridging cycle. What happens is calcium will go and bind to TNC subunits, as we discussed. It will cause conformational change. And I just want to point out that whenever anything binds to a protein, a receptor, an enzyme, it will cause a conformational change. The shape will change, and depending on the shape of the enzyme or the protein or the channel, whatever, it will affect its function and its binding ability to other molecules. Okay, so it all depends on the conformational changes. Okay, so at resting state, we had, for example, one position, but once calcium bound and it causes a shape change, we have another position. Okay, now, where was I? Yeah, okay, calcium will bind to the TNC subunit of troponin. Now it will cause conformational change. What happens is that it will cause the tropomyosin to move away from the myosin binding site. Okay, let me just go back a lot here. Okay, so let's pretend that this pointer is the calcium. Okay, calcium will go and bind to troponin, which is this purple circle. Okay, it will cause conformational change, and it will cause this tropomyosin, which is the red line, to move. Okay, it will move and it will expose the myosin binding site. Okay, so now myosin can bind to actin. Okay, this is the first step. Okay, let's go back. Yeah, so, okay, so yeah, we're done from that. Let's go here. Now, the myosin binding site is exposed, and myosin can bind. But how does it bind actually? Okay, so remember in the resting state, the myosin was in at a low energy state. It is inactive basically. Once the myosin binding site on the actin is exposed, the myosin will undergo conformational change. Okay, and ATPase, remember in the head of myosin, it had ATPase enzyme, and we had a regulatory chain for this ATPase. This ATPase will hydrolyze ATP into ADP and inorganic phosphate. Oh, sorry, this is ADP, okay? Adenosine diphosphate. I will change it, okay? Sorry about this. But it will cause the release of ADP and inorganic phosphate. So ATP will get hydrolyzed by the action of ATPase into ADP and inorganic phosphate. Now, this will allow the myosin to bind to actin, and it will cause the pivoting of the thin filament or the thick filament towards the center of the sarcomere. And this is what we call the power stroke. Okay? I'm just saying this in words. I'm going to open my camera from my phone to explain it in like demonstrating fashion. Okay? But just let me continue this part. Again, we have the myosin. It now combines to actin because the myosin binding site on actin is exposed. Okay. Now myosin will undergo conformational change. The ATPase enzyme on the myosin molecule itself will cause the hydrolysis of ATP into ADP and inorganic phosphate. And now ADP and inorganic phosphate will allow this releases energy, and this allows for the power stroke. Okay. Now it will cause the shortening of the sarcomere. The sarcomere itself will get closer to, to the center. And this is what we call the muscle contraction. Okay. So this is the power stroke. Now, we released ATP and we released ADP and inorganic phosphate. Now they are out in the, in the cytoplasm. It's not anymore bound to the myosin. Okay. Now, the ATP binding site on the myosin is, exposed. It's free. It doesn't have ATP. It doesn't have ADP. It doesn't have inorganic phosphate. It's exposed. Now, what happens is that ATP will go and fill this place. It will cause another conformational change, and the myosin is no longer, bound to actin. It will get detached from actin. Okay. So I'm going to open my camera now, 1 second, and adjust it to explain it. Okay. Is it open? Host. Can you please allow for the other accounts to allow them to open their cameras?
Is it not allowed?
It didn't. Let me try because it's from my phone, not from the laptop.
It's working. Yeah, I can see. It's working.
You can see me? Okay.
Yeah.
Can you see me? My face? Okay.
Yep. Yes.
Let's go. Okay. Okay. Let me. This hand is the myosin, or like the myosin head, and this hand is the actin head. Okay, or like the thin filament. Now, calcium came and caused the myosin head, the myosin binding site to be exposed, and now myosin can go unbind. Now, what happens is that we have an ATP molecule here, which will get hydrolyzed into ADP and inorganic phosphate. Now, once this happens with the ATPase, the myosin head can bind. Okay, and ADP and inorganic phosphate will release energy and causes a power stroke. This is the power stroke. Okay, now once they do the power stroke, they release energy, they will get detached. What will get detached? The ADP and the inorganic phosphate. Okay, the ADP and inorganic phosphate will get detached, the ATP binding site is exposed, and what happens is that another ATP molecule will come, like this is an ATP molecule, and it will cause a conformational change, causing the myosin head to detach from the actin. Okay, so again, ADP is, okay, this is ATP, okay, ATP. And I'm not now in the resting state, I'm not bound or anything. ATP will get hydrolyzed, and we have ADP and inorganic phosphate. And now I combined, and now the power stroke will happen. And then and then the ADP inorganic phosphate will leave. Another ATP will come, causes conformational changes, and I will get dissociated or detached from the actin. Okay, so again, myosin can bind to actin, causing a power stroke. ADP and inorganic phosphate will leave, and then another ATP molecule will come, causing conformational change, and now I get detached from the actin. Another power stroke. This is, the idea. Okay. Okay. Now, we already explained this. Yeah. Okay. Now, see here, this is another picture. A power stroke. See, this is the myosin head bound, and it went here. This is a power stroke. It will come back again, does a power stroke. Come back again, does a power stroke. And what happens is the sarcomere length will shorten, goes back to the center, or like goes closer to the center. Sorry, excuse me, to the center, and this is what we call contraction. Okay, so why is it? Okay, this is also what I discussed. The reactivation of myosin is like the ATP. The ATPase will hydrolyze the ATP into ADP and inorganic phosphate. It will return to its original position and resetting the cycle. Okay, um, okay. The myosin will reattach if the actin of like the myosin binding site on the actin is still available. What does this entail? It tells us that we still have calcium in the cytoplasm, and calcium is still bound to troponin C, and this caused the troponin myosin to move, and the active site is basically exposed. Okay, so this is again the demonstrating picture of the of the cross bridging cycle. Okay, where's number one? Number one is here. Now we have myosin head with ADP and inorganic phosphate attached. It will cause a power stroke. Okay, so a power stroke. And then ADP and PI, or like inorganic phosphate, will leave. It's not attached anymore. And now this site is available for ATP to come and bind. ATP bound to myosin will cause conformational change, and this will get detached from the actin, and then we repeat the cycle. Okay? Okay. Now we are done talking about the contraction, but you're in a state that you're not contracting your muscles all the time, okay? But what happens if you want to relax your muscle after this, cross-bridging cycle? So again, we said the main initiator of everything is calcium, okay? So if you remove calcium, you will not have contraction. Okay, this is basically the rule of it. No calcium, no contraction, muscle will relax. What is the physiology behind it? You remember when calcium entered the cell in the very beginning, it was bound to troponin C, okay? And this caused conformational change to the tropomyosin, which was covering the actin binding site, but once calcium entered, it moved its position and exposed the binding site. Okay, why this happened? Because calcium. Okay, if we remove the calcium, tropomyosin will go back to its original position and will cover the myosin binding site. You will not have myosin binding to actin anymore, and thus you will have relaxation. Okay, so this is basically the idea. Okay, okay. Now, what causes this calcium to be removed from the cytoplasm? Okay, in the very beginning, we said that calcium will enter from outside of the cell, and it will cause calcium-induced calcium release from the sarcoplasm. Okay, so calcium will go out from the sarcoplasm into the cytoplasm. Now we want to get it back into the sarcoplasm, and why is that? Because the sarcoplasmic, or like the sarcoplasmic reticulum is a calcium storage. It either releases calcium or it stores calcium back inside. Take calcium out, take calcium from the cell cytoplasm and back into the sarcoplasmic reticulum. Okay, what does this is the Circa pump. And Circa, it stands for sarcoendoplasmic reticulum calcium ATPase pump. This is a very big name. And this is just the words. Let's do it with a diagram. Okay. Okay. Now we have calcium outside in the cytoplasm, and we want to put it inside of the sarcoplasmic reticulum. And what does this is a pump, which we call the Circa, or, if you want to be like an intellectual, you can say sarcoendoplasmic reticulum ATPase. Okay. From the name, you can have ATPase. That means that this channel or this pump has an ATPase activity. What does that mean? It will hydrolyze ATP into ADP and inorganic phosphate, releases energy. And why do we need the energy for? We need the energy for active transport. Remember, we have a lot of calcium inside the sarcoplasmic reticulum and a lot less calcium in the cytoplasm, okay? All in any state, by the way, even if the muscle is contracting and we actually have high calcium in the cytoplasm. Normally, calcium concentration in the sarcoplasmic reticulum is way, way, way higher at all times compared to the calcium outside and the cytoplasm. Okay, so that's why we need ATP for active transport, and this is done by the pump, which we call circ. So calcium from the cytoplasm will go into the sarcoplasmic reticulum through the circa by active transport. Okay, again, calcium from outside or the calcium from the cytoplasm will get pumped by active transport by circa, by active transport because we have an ATPase activity. Okay, now again, it's active transport because it's against its what happened? Okay, because it's against its concentration gradient, and that's why it's an active transport. But what happens if we now logically we have a lot of calcium outside? It can leak through leaky channels, through calcium leaky channels, or maybe through any channels that can cause calcium to leave. We need calcium to be arrested, otherwise you will have contractions happening at all times. Okay, we need this because if you have contraction happening at all times, that means you have like tetanus, for example, or you have suddenly when you are sitting relaxing and suddenly you have your arms moving suddenly, or like you twitches. These are twitches or myoclonic contractions. What happens is basically calcium from the sarcoplasmic reticulum went out and caused the muscle contraction. Okay, so we need a police or like a guy, which is a calcium binding protein. It prevents calcium from leaving the sarcoplasmic reticulum no matter what happens until we actually want calcium to go out. Okay, this protein or this calcium binding protein, it is called calsequestrin. So cal for calcium, sequestrin, it sequests or like it basically like a policeman that doesn't allow calcium to leave. Okay, so this is how relaxation happens. We remove the calcium, which is the initiator of contraction, and we put it or pump it back again into the sarcoplasmic reticulum. Sarcoplasmic reticulum is like the cell or like the prison cell for the calcium, and calsequestrin is like the policeman that doesn't allow calcium to escape this prison. Okay, and this slide is basically what I just explained in words. Okay, now another part, and we are going to talk about going to move to the next part of the lecture, which is muscle mechanics. This is still in the cell. Now remember, we said we need calcium to initiate the contraction. We did the contraction, etc., but we also need ATP, and ATP was mainly used in three locations. First, the power stroke, when we said that ATP needs to be hydrolyzed into ADP and organic phosphate, releases energy and causes this power stroke. Okay, this is the first location. The second location is that ATP, another ATP molecule, will come and bind to the myosin head to cause the the detachment of this myosin from actin when we want to repeat, the cross-bridging cycle. So this is the second location. The third location is what we just discussed. We need ATP in this part to pump calcium again back into the sarcoplasmic reticulum. So again, ATP is used in three locations. We need it in the power stroke, we need it in the detachment of myosin from actin, and we needed to pump calcium back into the sarcoplasmic reticulum. Okay, now where do we get this ATP from? We get it from four locations, and 3 out of 4 of them. Maybe you took this in Reno, but we're going to discuss them either way. Now, ATP sources, you have free ATP in the cytosol, just free ATP. This is the first source, like free currency here and there. This is like free ATP roaming around in the cytoplasm. Okay. And the second source is aerobic respiration, oxidative phosphorylation, and electron transport chain. This is, the entire thing, how we regenerate ATP from, mitochondria and stuff. This is small, but this is another source of ATP, which is oxidative phosphorylation and the electron transport chain. Okay. The third source, which happens mostly during exercises or like anaerobic activities like running, swimming, etc. Anaerobic respiration and glycolysis, and the pathway that generates lactate. Okay, this is another source of ATP. Now, the fourth source of ATP, which is very uncommon, but it's like in muscle builders, the people who take kerosene during working out and stuff, this is the keratin phosphate. Keratin phosphate is going to get converted or get broken down into keratin. So this phosphate is like a source of producing ATP. So ADP, which is adenosine diphosphate, is going to take this phosphate from phosphokeratin or keratin phosphate. It's going to produce ATP, which is adenosine triphosphate. That's why we get this extra اسمه إيه ده extra phosphate from, and keratin, which is the substance itself. Okay, so this is another source of ATP. See, we produced ATP here. Okay, I hope that's clear. And now let's go to the actual heavy part of this lecture, which needs a lot of concentration and a lot of physics, which is muscle mechanics. Okay, now let's talk about definitions first, which is defining everything. I'm going to explain it, don't worry, but I need your full concentration here. Okay, we have two types of contractions, which we call isometric and isotonic. Iso means the same, like yeah, I just means the same, like hyper means a lot and hyper means low. Okay, so iso means the same. Metric, metric systems like length, centimeters, meters. So metric is length. Isotonic, tonic means tension. Okay. Okay, let's go through it. Okay, so isometric contractions is when your muscle contracts and there is no change in length. Okay, this is the scientific definitions of it. What is, the slang of it, or like how do we explain it? Okay, it's when you do an exercise. This woman right here is holding a squat. She is not moving her muscles at all. But is her muscles contracting? Yes. Is she working? Is she exerting force? Yes, of course. She's working her, lower limbs and glutes and quadriceps and etc. Okay. So this is when you are working out and you are holding. So for example, holding a squat, holding a plank, people who are focusing on increasing their muscle mass by holding or contracting their biceps without moving or curling it anymore. Okay, so you are exerting force. This is work. Okay, just holding a 1 minute plank is like a disaster. Okay, so you are still exerting work. Your muscles are still contracting, but there is no change in length. Okay, they are just here. Okay, so if this glute is contracting right here, it will stay this position. There's no change in length. But is it working? Is it contracting? Yes, it is. Okay, so this is what we call isometric contraction. Isometric contraction, muscle is working, there is no movement, and there is no change in length. Okay? Now, isotonic, on the other hand, is there is change in length while contracting. So this diagram right here, this woman is biceps curling. She is curling her biceps to work her biceps. There is change in length. Okay, so right here, her biceps is shortened. If she relaxed her arms, or if she went to resting position, her arms or like her biceps are relaxed. Okay, this is isotonic contractions. There is shortening, there is movement, but the tension is constant. Okay, why is the tension constant? Because let's say she is holding a 1 kilogram dumbbell, okay, and she is curling her biceps for like 5 reps or something. The tension that she is creating, or the tension that the muscles are creating while contracting, is the same, because she, for example, didn't change the dumbbell into 10 kilograms or 5 kilograms. Okay, so in isotonic contraction, you're moving your muscles and contracting it. In isometric contractions, you are contracting your muscles, but you are not moving it. Okay, it's like a tongue twister right here, but I hope you get it. So isometric contraction is about holding something steady without movement, and that's why in isometric, same length, no change in length. Okay, isotonic is like lifting weights. There is same tension because you are lifting the same amount of weights, but you are still moving your muscles. Okay, like biceps curling, lunges, squatting, up and down, running. Okay, okay, okay. This is isotonic and isometric was the first set of definitions that we need to know. Now, the second set of definitions that we need to know is active tension and passive tension. Okay, so active tension, it is generated when the muscle contracts. What does that mean? Is that physiologically speaking, is that there is binding between between actin and myosin. There is cross bridging happening. There is active force happening, and tension is a type of force. So when I say active tension or active force, it's the same thing. Similarly, if I say passive tension or passive force, it's the same thing. Why? Because tension is a type of force. Okay, now active tension is when you are actually contracting your muscle, when you flex your biceps, when you feel the resistance of you lifting weights. When you are doing lunges and feeling your quadriceps contracting, this is active tension. And what does it depend on? It depends on the number of cross bridges being formed, because the physiology behind active tension is basically the cross bridges between actin and myosin. Okay, so if you, for example, let's give you an example. Your biceps curling, but you are curling it midway. Okay, not the entire curl. Okay, if you are bending it midway, that means you are not contracting it to the fullest. Okay, if you contract it to the fullest and curl the entire way up to your arms, that means your active tension increased because the number of cross bridges increased because you contracted your muscle more. Okay. Okay, so this is the what is active tension is all about. It is basically the amount of cross bridges happening between actin and myosin. And basically, if you increase the cross bridges, you will increase the active tension. Okay. Now, the passive tension, on the other hand, is the resistance you feel when you are stretching your muscle but not contracting it. Okay, so this lady right here is stretching her hamstrings. Um, Please, you can stretch your arms right now and you can feel resistance. You cannot stretch for example, if you are not trained to do a split, you cannot exceed the level of that your body is adjusted to, right? This basically is what passive tension. تمام. So passive tension is when you are stretching your muscle but not contracting it, but you are still doing work. Stretching requires a lot of work as well. And what is the physiology behind passive tension? It is the elastic properties of the muscle fiber and the connective tissue around it. Okay, so the muscle is an elastic tissue, and any elastic or soft tissue, it has connective tissue around it to support it. So this is the elastic property of the muscle, is the passive tension. Okay, so again, active tension is when the muscle contracts because of the cross bridges between actin and myosin. Passive tension is when you are stretching your muscle but not contracting it due to the elastic properties of the muscle and also the connective tissue around it. Okay, now, okay, the third set of definitions is the length of the muscle. Okay, so you have the normal resting state of the muscle. It's called the optimal length. Okay, you are not contracting your muscle, you are not shortening it, you are not lengthening it. So you're not shortening it by, for example, you're not doing a bicep curl, or you're not contracting your muscle, and you're not lengthening your muscle when, for example, you are stretching it. So the optimal length or the resting length of a muscle is basically the length at which the muscle can exert its maximum amount of force. Why? Because there will be optimal overlap between actin and myosin. I'm going to show you a diagram of it, but just bear with me for a moment. Okay, so the optimal length of the muscle is what? Is the resting length. Why is that? Because it will be it's the length at which the muscle will generate the maximum amount of force, so the maximum amount of active tension, and there will be an optimal overlap or maximum overlap between actin and myosin. Okay? Now you are doing a biceps curls. Okay? This is a shortened muscle because you are contracting it. The muscle is shortened. Okay? Happens is that there will be a limited room for the filaments to slide because you already contracted your muscle. The sarcomere length is already shortened. It will not be shortened beyond this length. Okay, you cannot shorten for example, you can shorten your muscle to 5 centimeters, and this is the maximum amount that you can shorten your muscle. This is the maximum amount. You cannot shorten it, for example, to 4 or 3. Okay, it cannot exceed this length. So this is the shortened muscle. There will be limited room for filaments to slide, or there's maximum amount of what do you call it, actin myosin cross bridges. You cannot shorten the muscle beyond this length. Okay. Now, lengthened muscle, on the other hand, is when you cannot stretch the muscle beyond this length. Okay, it's the physiology behind that is that there is the largest distance between myosin and actin filaments. Okay, there will be less overlap between actin and myosin, and less force can be generated. Okay, so let's say you are doing. Let's go here. This lady is doing a hamstring stretch. Okay, can she, and the muscle length is the maximum. See, she is stretching her muscle. Can she, for example, go up suddenly and do like a curl for her hamstrings? No, she cannot do that unless she resets her position. Okay, why? Because the muscle is it's stretched beyond its limit. And the second thing is that there is the distances between the filaments is the largest. When you are stretching the muscle, you are increasing the length of the muscle, you are basically pulling the filaments away from each other. Okay, so optimal length, optimal overlap, greatest force. Shortened length is all the filaments are overlapped. There is less force because you cannot. Where is the force? You're already contracting muscle. And lengthened muscle is less overlap. There is less force because you stretch the muscle. Okay. Another word for shortened length is below the optimal length or below the resting length of the muscle. And another word for lengthened muscle is above the optimal length when you are stretching the muscle. Okay, now graphs. Okay, because what I just discussed were definitions and explaining the meaning of these words. Okay, now let's apply them with graphs. Okay, now this blue line, if you can guess, before I actually explain, this line depends on like it's describing which length of the muscle. It describes the optimal length of the muscle. Okay, why? Because it is the optimal length or the resting length of the muscle is the length at which the muscle can generate the maximum amount of force. Okay, so this is here. This side will be below the optimal length, and this side will be above the optimal length. Okay, so here you are shortening length or below the optimal length. Here you are stretching the muscle, the lengthening muscle. Okay, now as we said in previous slides, active tension is the force or tension generated when the myosin and actin are cross-bridging one another. And active tension, as you can see from the graph, this is the black line is the active tension forces. It's the maximum at which length? At the optimal length. So active tension is maximum at the optimal length of the muscle. Why? Because at the optimal length, you can generate the maximum amount of overlap between the actin and the myosin. And when you do the maximum amount of overlap between actin and myosin, you have the maximum active tension. Okay, so let's say here at the shortened length of the muscle is shortened. There is limited amount of overlap that can happen. I mean, I'm talking about new overlaps that can happen. Okay, so there is less force that can be regenerated, and there is less active tension. Okay, on the other hand, above the optimal length, which is the blue line from here, the filaments are very further away from each other because basically the muscle is stretched, right? Because here it should be the lengthened muscle or the lengthened length. Okay, um, okay, so this is the muscle will be stretched here. The filaments will be further away from each other. You have less cross bridging happening because the distance is away. They are further away from each other. So what happens? Less cross bridging can occur and less active tension, and that's why the active tension is optimal at or like maximum at the optimal length of the muscle, which is what the resting state. Okay, now for the passive tension. Passive tension, again, is the tension or the force being generated when you are stretching the muscle. That definitely means that when you increase the length of the muscle by stretching it, you will have more passive tension. And that's why as you stretch the muscle or as you increase the length of it, the passive tension increases. Okay, so again, to summarize before going to the next paragraph, the optimal length is the maximum or the length of the muscle at its resting state where you have a maximum active tension being generated. Why? Because you have the maximum amount of cross-bridging that can occur. Okay? Below the optimal length or above the optimal length, the active tension will decrease. Okay? The passive tension is the tension generated or the force generated when you are increasing the length of the muscle or when you are stretching the muscle. And that's why as you increase the length of the muscle, the passive tension will increase. Tamam? Okay, I hope that makes sense. Now let's go to this graph. Again, this is basically what we explained, but in more, pictures and more, and in more explanations. Here you have the sarcomere. Okay, this is one sarcomere, this is one sarcomere, and it's basically telling you the overlapping length between these two. So here, at the very, very shortened length of the muscle is very, very contracted here. Okay, let's say it's a very stiff muscle, or you are contracting your biceps at the maximum you can. Okay, the cross bridges are already formed. Yes, I agree with that, but will there be new cross bridges forming? No, because there are no free spaces. The muscle is very short here. So this is shortened length. Tamam? Okay, now this length here is the resting length of the muscle. Very amount of overlaps can happen here. The muscle can lengthen, it can shorten. It's the optimal length and it's the resting length. So right now I am not contracting my muscles. I can generate the maximum amount of force if I decide to contract it or if I decide to stretch it. Tamam? Now, as you increase the length of the muscle, let's say I am stretching my hamstrings, okay? The length between the thin filament and the thick filament is increasing. Will cross bridges happen here? No. Why? Because the distance itself is like they are being stretched, like pulling away from each other. Okay? So the dashed line, which is this line here, is the active force or the active tension. Again, I said active force or active tension is the same thing because tension is a type of force. Okay? So at the resting length or at the optimal length, active tension is the is at its maximum, magnitude. And as you increase the length of the muscle, the passive force or the passive tension increases, because the passive force or tension is the force at which the muscle is stretched. Okay. Now, total force. What is total force or what is total tension? It is basically active tension or active force plus passive tension or passive force. Okay. Um, Okay, so let's give you a number to better understand it. Let's say here it's 5. Okay, let's say here it's 5, and let's say here is 10, or like here is 5. Here it's 5. So active tension is 5 and passive tension is 5 at this length. So 5 plus 5 is 10. So total force will be 10. Okay. Now, some people in your recordings asked, why do we have a decrease in this total force? Shouldn't it increase because you are increasing the active force? And actually, that's a very smart question. But the answer to that question is, now you are beyond the optimal length. Okay. And we said that the active tension or the active force will decrease beyond the optimal length. Okay. Because the active force is decreasing, the total force will decrease. So let's say here this was 5 and this became a 3. Okay. And let's say here it's still 5. Okay. So 5 plus 5 is 10. So here it's 10. And 3 plus like I don't know, 7 or something, it's going to be decreased because the active force is decreasing. Okay. Um, this is what I just explained in words. Like, we can go through them again, but literally what I just explained in these two diagrams, and in words. Okay, you can read them, and the highlighted things or the bolded things are are things to focus about. Okay. Okay, now for another concept, which is the force velocity concept. And I'm going to explain this with a practical example. Okay, so you are a very healthy person and you work out every day, mashallah, and you lift weights, okay? But at the very beginning of your fitness journey, you started with no weights, and then you started increasing the weights a bit, okay? So let's say you can lift 10 kilograms is your maximum, but you also work out with 5 kilograms. Generally speaking, when you lift 5 kilograms, you contract or you move or you have more amount of reps of the whatever exercise you're doing, more than the 10 kilograms. So let's say you can do 20 reps of biceps curls with a 5 kilogram, but you can only do 10 reps with 10 kilograms. Why is that? Because the load on the muscle is more with the 10 kilograms, obviously. And the load on the muscle with the 5 kilograms is less compared to you because you're a very healthy person. Okay? Now, while you are contracting the biceps with the 5 kilograms, you are contracting it very quickly, because it's very light on you. That is basically the velocity of the muscle contracting. Okay, so as you increase, basically this graph, what it explains is, as you increase the load, so from 5 kilograms to 10 kilograms, the velocity or the velocity of the muscle contracting or the shortening of the muscle is what is decreasing. Okay, so you are having very difficult time contracting your biceps with the 10 kilograms. It takes you very a long time because your velocity is decreasing because you increase the load. Okay, so the relationship between the load or the force you are exerting on the muscle is inversely proportional to the velocity of the muscle, and Vmax is the maximum velocity, basically, just contracting it very quickly. Okay. Okay, now we are almost done with skeletal muscle. I know it took a long time, but inshallah, it's going to be easy. Now, what affects the ability for a muscle to contract? Obviously, a duration of action potential. Now, what started the entire thing from the very beginning is the depolarization wave. Yes, so the action potential, so there's an action potential. After that, will cause a muscle contraction. One wave of action potential will cause one wave of muscle contraction. Okay, so how does this impact the contraction? Why does an action potential impact the contraction of a muscle? Because when a depolarization wave comes, it will go through the TT rules, it will cause calcium release, calcium will go bind to troponin, cross-bridging cycle happen. Amen? So let's work out a bit. Now, action potential, if you have higher or more frequency of action potentials, you will have basically more calcium being released or a faster rate of calcium released, and you will have more prolonged contraction because you are contracting very fast, okay? Prolonged as means like 10 contractions, for example. So if you have 10 potential waves, you have 10 contractions. But if you have 5 action potential waves, you have 5 contractions, in simple terms. Okay. Now, this is the normal case. Again, one action potential wave will cause one contraction. One action potential wave will cause one contraction. If you notice here, we have the same frequency of the action potential, and we have the same frequency and the same force, which is the magnitude. This is the magnitude of the muscle contraction. Okay, this is what? This is synchronized contraction. Alhamdulillah, there is no altered electrical activity of the muscle, so you don't have twitches, you don't have tetanus, for example. Okay, because this is what? This is the normal case. One action potential wave, one muscle contraction. One action potential wave, one muscle contraction. Tamam? With normal frequency and normal magnitude. Tamam. Okay, this is the normal case. Now, for the summation phase or like fused twitches, it's still, you have normal action potential waves with normal frequency, but one contraction adds up to the next contraction, adds up to the next contraction. Okay? Tamam? In this diagram, it is the same, but it's because we increase the frequency of the action potential waves, see, like more action potential waves, you will have fused twitches because they will sum together, they will be added together. Okay, you still have one wave after another with the same frequency, but you are adding or you are summing each individual contraction. So this is one contraction, this is one contraction, this is one contraction, you are summing them. Tamam? Here it's the same case, but because less frequency of action potentials. Tamam? Okay, now here it's in the pathological state, now like in tetanus. Okay, this is at very, very high frequencies of the action potential. There's no even space. Like, look here, it's 100 hertz. Each individual contraction, which should happen, is now one entire contraction, and this is what we call a tetanic contraction or tetanus. Okay, which is obviously pathological. Tamam? So it all depends on what? It all depends on the frequency of the action potential. Tamam? Okay, now this is a muscle fiber, and this is a neuron activating or supplying or innervating this muscle fiber. Now, this is what we call a motor unit. So what is a motor unit? A motor unit is one muscle with a motor nerve supplying it. So a motor unit is one muscle supplying it. So if you have more neurons supplying one muscle fiber, you will have more contractions because you have increased supply to the muscle. Why do we need to know this? For research and experimental reasons, but it's actually applicable in our body. So if you have more muscle fibers, let's say the fibers or the number of fibers of your quadriceps, for example, is more than the muscle fibers of your hands. Okay, that's just normal anatomy of it. What does that entail? It entails that you can contract or generate more force with your lower limbs compared to your upper limbs, and that's why lower body exercises is easier. That's why you can walk without getting tired. Why? Because there's a recruitment of neurons to this specific muscle fiber, a lot of neurons supplying one set of muscle. Tamam? Okay. Again, this is just what I just said in a graph and in a diagram. The more muscle, the more neurons is being recruited to or innervating a muscle, the more action potential or force it can generate. Tamam? Okay, now for questions, and we can take like a 10-minute break. Okay, so a 16-year-old adolescent boy on a track team asks his pediatrician if he can take creatine on a regular basis in order to increase his muscle strength prior to track meet. Which of the following most likely explains why he wants to take creatine? So I'm gonna wait for like 20 seconds to think about it, and then I'm gonna explain the answer. Okay? Okay, so the answer is D. Okay. You might think, why is this question related to here? I'm going to tell you, it's like a low yield question. Tamam? Remember when we talked about the sources of ATP? When we finished talking about the muscle contraction, we said we have ATP from 4 sources. What are they? The free ATP in the cytosol. The other source is from oxidative phosphorylation and the electron transport chain. The third source was from anaerobic respiration, and the fourth source was from phosphokeratin. So that's why the answer is D. Okay, so you are taking creatine because in the muscle it will be converted into creatine or phosphoryl creatine. And when you need this energy, it will be broken down. This phosphokeratin or keratin phosphate will be broken down to give you ATP. Okay. 1 second. I'm gonna show you the equation. It's a little bit up here. I think it's here. Yeah. This is the equation I'm talking about, and this is basically the answer of the question. Okay. Let's go back. تمام. Okay, this is another question. A 24-year-old medical student is avid bodybuilder who lifts weight approximately 2 to 3 hours per day. Which of the following best describes the action potential of skeletal muscle during his workouts? So again, I'm going to wait for 20 seconds and then I'm going to explain the answer. Okay, so the answer is E. Okay, action potential. To reach the skeletal muscle at the neuromuscular junction, the depolarization wave should go through the invaginations of the cell membrane, which is which causes the depolarization opening of calcium channels and starts the entire thing that we just discussed. تمام? Okay, now this is the third question and the last question in this part of the lecture. And okay, which of the following best describes the contractile response of a skeletal muscle? Okay, so this is the answer. More tension is produced when the muscle contracts isometrically than isotonically. Okay, let's go through this question bit by bit and rule out the other responses. So more tension is produced. Why? Because in isometric contractions, in isometric contractions, you have increased tension, but you are not changing the length. You are working your muscles. You are holding a squat, for example. You are working your glutes. This is very stressful. It's very hard. You are creating a lot of tension, but there's no muscle movement. Okay. In isotonic, it's even from the word itself. Isotonic, there is the same tension, so there will be no change in tension during isotonic contraction. تمام؟ Okay. Muscle contraction starts after the action potential is over. No, because we discussed that after an action potential wave, you have a muscle contraction. One action potential wave, one contraction. تمام? In muscle length decreases to a greater extent in isometric than in isotonic. No, why? Because in isometric there is no change in length أصلا. تمام? Okay. In repeated stimulation, the repeated stimulation decreases the magnitude of the muscle contraction. The stimulation, it doesn't depend on the force of the muscle contraction. It depends on the number of muscle contractions happening. So you are not creating more force when you are stimulating the muscle. You are just increasing the number of contractions. So instead of doing 5 biceps curls, you are doing 10 biceps curls. Okay, the duration of the action potential exceeds the duration of skeletal muscle contraction. Again, they should be synchronized in a normal state, so that's why we rule out this question. Okay. Okay, now we can take a 5 minutes break because I really need to drink some water and then we can continue. Okay, so host, should we stop the recording or? Hello? Okay, let's continue, inshallah. Just give me a moment. Okay, so we're going to talk about small muscle contraction. Inshallah, it's not going to take a lot of time. It's just, the basic differences between the skeletal muscle and the small muscle. But let's talk about it. Okay, so again, the initiator of any type of contraction in any contractile tissue is calcium. Okay, for smooth muscle, where do we get calcium? We get it again from extracellular matrix or extracellular space and from the sarcoplasmic reticulum. This image here. Okay, so this is the sarcoplasmic reticulum, and this is the calcium channels. Calcium will go out from the sarcoplasmic reticulum into the cytoplasm. تمام? This is from the cytoplasm, the source of calcium. Again, it's in the cytosol. Now, from the extracellular matrix, or the space outside of the cell into the cell, we have actually three types of channels, but I just drew two here. We have the ligand-gated channels, where we have a ligand, which is a hormone, a neurotransmitter, a drug, an ion. Anything that binds to a ligand-gated channel will open the channel and cause calcium to enter. So it's the calcium ligand-gated channel, or like a ligand-gated calcium channel. Another channel, or another type of calcium channel in the smooth muscle, is the voltage-gated calcium channel. It will sense changes in the membrane potential, or it will sense changes in the voltage. And if it reaches the threshold where it opens, it will open and cause calcium to enter. Why? Again, because generally in any cell, we have more calcium outside than inside, and more calcium in the sarcoplasmic reticulum inside than the cytosol. Okay, so that's why calcium will go down its concentration gradient from both directions. Tamam? Okay, now the three types of actual calcium channels is, we said, ligand-gated, voltage-gated, and mechanically gated. Ligand-gated channels will open as a result of a hormone, a neurotransmitter, a drug, an ion, any ligand. Okay, a voltage-gated channel will open once it senses a membrane potential, once it senses a voltage. A mechanically gated channels, it senses mechanical stimulus, like what? Stretching. Okay, so these are the three types of calcium channels found in the cell membrane, and the other type of calcium channel in the sarcoplasmic reticulum will cause calcium release into the cytosol. تمام. Now, more about the ligand-gated channels is that once, let's say acetylcholine, because you guys know what acetylcholine is. Acetylcholine binds to a ligand-gated channel, and it causes the activation of G protein. And in this case, it will activate GQ, because why GQ? Because it will activate the IP3 DAC pathway. I'm not really sure if you took the cell signaling pathways in one, but if we want to stimulate IP3 and DAC pathways, we will stimulate the GQ protein. Okay, this is like a general rule. تمام. So acetylcholine will go bind, it will stimulate G protein, GQ specifically. It will convert the phospholipase C into IP3 and DAG. It's not in the image, but it will also become DAG. And IP3 will go and stimulate or activate calcium channels in the cytoplasm, and then it will cause calcium release. تمام? Or calcium efflux from the cytoplasm into the cytosol. تمام? And once calcium, again, the same thing in smooth muscle as in skeletal muscle. Once calcium is in the cytoplasm, it will cause or initiate contraction. تمام? So this is a smooth muscle which is relaxed, and we want it to contract. What are the steps? Again, calcium entry, which we discussed, either from the extracellular matrix through three channels: voltage-gated, ligand-gated, or mechanically gated. تمام? Or from the sarcoplasm, from like 3 DAC pathway, or calcium-induced calcium release, which is the same steps as we discussed in skeletal muscle. تمام? Myosin receptor is opening and causes calcium release. Now, the second step is calcium calmodulin complex formation. Now, bear with me here because it's a tongue twister. I'm going to call them the CC complex, calcium and calmodulin. What is calmodulin? It is a calcium binding protein. It's just a protein that binds to calcium. And from the name, we can dissect the name: cal from calcium, modulin from modulating. So it regulates calcium or modulates calcium. Okay, this is the second step in our smooth muscle contraction. So calcium enters, calcium goes and binds to calmodulin and forms the CC complex, or calcium calmodulin complex. Then the second step, which is the activation of this MLCK. MLCK, what does it stand for? Myosin light chain kinase. Okay. what? Sorry about that. Okay, myosin light chain kinase. Very from the very beginning of the lecture, myosin had four light chains. تمام? And these light chains has to be regulated, and what regulates them is two enzymes, and one of them is the myosin light chain kinase. And a rule in everything, a kinase will add a phosphate to something, a phosphatase will remove a phosphate from something. So in a case of myosin light chain kinase, it will add phosphate to the myosin light chain, and it will cause what? You guessed it, it will phosphorylate this light chain. And when something is phosphorylated, generally it will be active. It will be activated. تمام? So MLCK will be activated by what? What activates this enzyme is the CC complex. تمام? So again, calcium will enter, calcium will bind to calmodulin, forming the CC complex. The CC complex will go and activate the MLCK. As well as MLCK, it will go and cause the phosphorylation of the light chain of myosin, and it will cause conformational change to the myosin light chain. It will allow the myosin to bind to actin, forming cross bridges, forming the power stroke, forming contraction. Okay, this bit right here is the same exact thing that we talked about in skeletal muscle. تمام? So it's only the differences in the prior steps to it. So in skeletal muscle, we just had calcium coming and binding to troponin. In this case, calcium goes and binds to calmodulin, goes and activates an enzyme, which is a kinase. This kinase will phosphorylate the myosin light chain, and it causes, the cross bridging and the power stroke. تمام? Okay, the next step. Okay, now see what I did here is that the contracted muscle, we want to relax it again. Okay, so here the muscle was relaxed, now it's here it is contracted and we want to relax it again. Okay, so in smooth muscles, we want to okay, what caused the contraction in the first place? It's the phosphorylation of the myosin light chain by myosin light chain kinase. تمام? Now, in this case, we will dephosphorylate the myosin light chain. We will change the shape of the myosin head, or like the myosin light chain, and it will get detached from the actin. تمام? So what does this, what controls this process, or what controls this step is the myosin light chain phosphatase. A phosphatase, as I mentioned, will remove a phosphate from something, and generally speaking, when you remove a phosphate from something, you deactivate. You deactivate the something that you dephosphorylated. So when you want to relax a smooth muscle, the myosin light chain phosphatase will dephosphorylate the myosin light chain, causing conformational changes. It will detach from the myosin, from the actin head, and then you have relaxation of the smooth muscle. So speaking generally, or like generally speaking, the smooth muscle contraction depends between the balance between the myosin light chain kinase, the kinase, which adds phosphate, and the myosin light chain phosphatase, which removes the phosphate. Okay? Okay, now this is what we just explained in a picture. Again, calcium will enter the cell because of depolarization or because of a channel opening or sarcoplasm released calcium. Anyways, calcium is in the cell. Okay, calcium will bind to calmodulin, forming what? The CC complex or the calcium calmodulin complex. Calcium calmodulin complex will go and activate MLCK, or like myosin light chain kinase. Kinase will what? Will add phosphate to the myosin. And when you add phosphate to something, you activate it, cross bridges happen. Okay, now you want to relax the smooth muscle. What happens is MLCP, or myosin light chain phosphatase, will come and remove the phosphate. See, myosin here doesn't have a phosphate. When you remove a phosphate from something, you deactivate it, and thus you have relaxation. Okay, ignore this part, we're going to discuss it later. تمام? Again, this is the picture from your lecture. Inactive myosin light chain, calcium calmodulin complex, تمام? Okay, now we want to regulate the smooth muscle contraction. Okay, as I said, it is the balance between myosin light chain kinase and the myosin light chain phosphatase. تمام? It is all about balance between these two enzymes. So you either enhance this enzyme or decrease its activity, or you enhance this enzyme or decrease its activity. Okay, so if you enhance the activity of the kinase enzyme, it will it depends on the calcium calmodulin complex. So in other words, it depends on the concentration of the calcium. If you have high calcium, you will have higher activation because you have higher complexes being formed. In higher activation, you will have higher kinase activity. In higher kinase activity, you have more contractions. تمام. Okay, now if you have low calcium and hypoglycemia, calcemia, for example, less activation of the kinase enzyme because you have less CC complexes being formed. If you have less activation, you have less activity. In less activity, you have less contraction. Okay, again, a balance means equilibrium. So if you have balancing sides, you have the MLCK and the MLCP. Okay, if you decrease the MLCK, what will increase? The MLCP, and vice versa. Okay, so it is basically an equilibrium, and if you decrease the activity of MLCK, the equilibrium will shift or favors the MLCP. Okay, basically you are having less contraction, the muscle is relaxing. تمام. Okay, if you have higher MLCK, this is the rule of thumb, higher MLCK, you have contraction. Okay, another very important thing to note is that if this enzyme is phosphorylated, so the MLCK itself has a phosphate on it, it has less affinity to the CC complex. Okay, so I have the MLCK and it is normal, the CC complex will go bind and activate it. Okay, but if this MLCK is phosphorylated, it has a phosphate group on it, it has less affinity or less binding capacity to this CC complex. تمام. If it has less affinity, it will bind less to the CC complex. The CC complex is responsible for activating it. So if it doesn't bind, it will not be activated, and you have less contraction. تمام. So this is another rule. In MLCK, activity can be decreased by two things. If you have low calcium, which is like, it makes sense because you have less CC complex formation, or if the MLCK itself is a phosphorylated MLCK and it has a phosphate group. And why? Because it has less binding capacity or less affinity to the CC complex. Tamam? So how does this MLCK gets phosphorylated in the first place? Is because of any cell signaling pathway that induces cyclic AMP and PKA pathway. Okay, anything which stimulates the cyclic AMP PKA pathway. PKA is a phosphokinase A. Phosphokinase A will go and phosphorylate MLCK. Phosphorylated MLCK has less affinity to CC calcium. So I know it's a tongue twister, but go through the steps one by one. So I will repeat it again to make sure that you get it. MLCK is responsible for activating the myosin or phosphorylating the myosin to allow for cross bridges. MLCK gets activated by CC complex. تمام? MLCK gets activated by CC complex. This depends on the calcium concentration. So if you have low calcium concentration, you have less CC complex formation, you have less activation of MLCK. تمام? This is the first step. The second step, if any secondary pathway that stimulates the cyclic AMP and the PKA pathway, anything which stimulates that will go and phosphorylate the MLCK enzyme and phosphorylate it. So phosphorylated MLCK enzyme as a structural protein is less, it has less affinity to the CC complex. If it has less affinity to CC complex and it will not bind, it will not be activated, it will not cause the phosphorylation of myosin and no cross bridges. Okay. Now MLCP, on the other hand, which is the myosin light chain phosphatase, is responsible for muscle relaxation. تمام? So if you have low calcium and if you have low CC complex, the equilibrium will shift or favors the MLCP. The same thing, if you have phosphorylated MLCK, you have less contraction or activation of the myosin. It will favor what? It will favor MLCP. Why? Because it is an equilibrium. تمام? Now, this is these two steps is basically when the MLCK itself is low or when the calcium is low and it favors MLCP. But is there something which activates MLCP? Yes, there is. So we have the nitric oxide, and if you guys know from your previous blogs, nitric oxide is a vasodilator. It relaxes a muscle, and this is how it works. This is the physiology behind it. So the nitric oxide will go and stimulate the pathway of JTP or cyclic GMP, and it forms the cyclic GMP. And cyclic GMP, PKG, which is pathway, it's the pathway like here. Cyclic AMP PKA pathway. Here it's cyclic GMP PKG pathway, so phosphokinase G pathway. This pathway will enhance the activity of the MLCP. تمام? So just to memorize it, nitric oxide will stimulate the cyclic GMP pathway, which will activate MLCP. If you have high MLCP, you will dephosphorylate the myosin head, you will cause muscle relaxation. Then this is what I just said. In pictures, nitric oxide, GTP or cyclic GMP pathway will stimulate the MLCP. MLCP will remove the phosphate from the myosin. This myosin doesn't have a phosphate, and it will cause relaxation. تمام? Okay. We are done from how the muscle itself contracts. Let's see how does it relax. We already said it's a balance between MLCP and MLCK, but also it is about calcium. Calcium initiates everything. So we want to relax the muscle completely. We don't want calcium to bind to calmodulin, and we don't want the contraction to happen. What we need to do is we need to put calcium inside the sarcoplasmic reticulum again, or kick it out of the cell entirely. So if you want to kick it out of the cell, we have two channels, either the calcium sodium antiporter, so calcium will be kicked out in exchange of sodium, or the calcium proton, which is, this is a proton antiporter. So you'll get the proton in and you'll kick the calcium out. So these are the two channels, or the antiporter on the cell membrane. This is the cell membrane, okay, out of the cell. Or the other approach is pumping calcium back into the sarcoplasmic reticulum. Circa, again, which we discussed, Circa will pump calcium against its concentration gradient using ATP because it has an ATPase activity to it. And calcium, once inside the sarcoplasm, will be arrested with two proteins, which is, one is calsequestrin, which we already talked about, and the other protein is called the calreticulin. Okay, it's just calreticulin. So we have two proteins, calsequestrin and calreticulin. But just know calsequestrin because it's more asked about. But basically, what are they? These are two policemen that are arresting calcium inside the sarcoplasmic reticulum and doesn't allow it to leave. تمام? Okay, so these are the three ways which calcium can be kicked out of the cell or into the cytoplasmic reticulum, preventing calcium to binding to calmodulin, preventing the contraction of the muscle altogether. Okay, now again for the mechanics part of it. Okay, so we have two types of contraction in a smooth muscle. We have phasic and tonic contractions. تمام? Okay, so the phasic contraction of the muscle is in phases, it's in rhythms, okay? It's a synchronized contraction in waves, okay? This is basically the idea of it. So the phasic contraction is a rhythmatic contraction, and it helps us because, in the small intestines, in the GIT, in the urinary bladder, in the uterus, we want this rhythmic contractions, and it helps the function. So in the smooth muscles in peristalsis, even esophagus in peristalsis. Okay, it's a rhythmic contraction, one contraction after another in a rhythmic state by a smooth muscle is called a phasic contraction. Right? In the urinary bladder, when your bladder constricts or contracts when you are micturating, and in the uterus during labor, we need rhythmic contractions of the uterus to induce labor. Okay. This phasic contraction is regulated by two things: hormonal control, like in the uterus, and then you took in endocrine, like oxytocin and estrogen, okay? And in neural control is basically the neural fibers that are recruited in this area, تمام? And then the phasic contraction, it is short duration. So you have one contraction after the other in a rhythmic state, but the contraction itself is short, okay? So like one contraction after another in a rhythmic motion. Okay, this is the graphic representation of what I explained. Okay, this red line is for calcium concentration. Ignore the green line, we're not going to discuss it, but observe the force, or like the purple line. Let's discuss it now. Okay, so you have high calcium concentration and it will induce contraction. It will induce contraction to have increased force or increased contraction of the muscle, but after the calcium is done, because it was kicked out of the cell or it's pumped back into the sarcoplasmic reticulum, the contraction will stop. This is phasic contraction. It's a rhythmic contraction, one contraction after the other in a rhythmic state. تمام. And it depends on the calcium concentration. I mean, all of them depends on the calcium concentration, but once the calcium concentration is constant, the force will be constant, and we will not have another contraction unless you have another rise in calcium here. The force will increase. Right? Okay. The other type of contraction of a smooth muscle, these are basically properties of a smooth muscle, is a tonic contraction. So tonic contraction is when the contraction or when the tension is sustained for a long period of time without you using ATP, without you using energy. Okay, so the muscle is contracting for a long time with minimal amount of energy. And why is that important? In regulation of blood vessels, for example, in blood pressure, in blood pressure by regulating the muscular tone. تمام? Or when you are in a resting state, you are basically still generating urine, and the bladder is not directly, contracting and you are micturating. Yeah, the bladder is, filling itself, and it is still it is, stretching or like increasing in its size by tonic contraction. تمام. So the tonic contraction of a smooth muscle is just a property. It's like a character, a character of a smooth muscle, or like the way the fibers are arranged, because it allows us to contract the muscle without using energy, or like without using a lot of energy. تمام. Okay, so the physiology behind it is because you have a lot of contraction. Contraction depends on the MLCK, followed by slower rate of myosin dephosphorylation, so less rate or like less activity of MLCP. So the activity of MLCK higher than MLCP, that means that the smooth muscle is undergoing tonic contractions. تمام. Another name for tonic contraction is latch. So if you found in questions latch, latch contractions or tonic contractions, these are basically the same thing. What does that mean? It means that the muscle, as a character or like as a property of the smooth muscle, it can contract for a prolonged period of time without using or without using or wasting a lot of energy. تمام. So imagine if you are constantly regulating your blood pressure because you are changing positions and etc., and the muscle is always using energy, because if you took one step or like 5 steps, you will be tired because you are using a lot of energy. تمام. So this is a very good property to sustain your energy expenditure. Okay, this is what I just discussed in a graph, and okay, you can definitely see the difference. Okay, you have calcium or you have contraction. When you have calcium, you have contraction. So calcium is high, you had contraction, and now it's getting lower. But in tonic contractions of a smooth muscle, it can sustain the contraction. The force here is a contraction with low calcium, and for a prolonged period of time. So you are not using energy. You are increasing or the time of the contraction without using a lot of energy. So this is basically tonic or latch contractions. تمام? Okay, this is basically, you can read this. It's like the differences between skeletal muscle contraction. In the regulation in skeletal muscle, it was only calcium, but in the smooth muscle, it was the calcium is from the action potential, but here calcium can be from neurotransmitters, hormones, stretch, the calcium channels that we discussed. In skeletal muscle, it was the dihydropyrimidine and the ryanodine receptors because of the action potential or the depolarization wave. In smooth muscles, it was the voltage gated, and the mechanically gated channels. In the smooth muscles, you have slower contractions for a prolonged period of time, which we just discussed, which is the last day, latch contractions. Okay. And in smooth muscles, you don't have a lot of sarcomeres compared to skeletal muscles for obvious reasons, because that means that smooth muscles can be stiff, and we don't want that. We want smooth muscles to be flexible, to accommodate, the fluids, the food, the blood, etc. Okay. Again, this is the differences between them in mechanics terms. Again, we discussed this graph before. You have the active tension, the passive tension, and the total tension or the total force. Active tension is optimal at its optimal length, and the passive tension increases as you increase the stretching of the muscle. This is in case of what? Skeletal muscle. In case of smooth muscle, you can already see the differences between the graphs in terms of the size of the curves. Okay, so you have this curve here, which is the active force. It is still active force is maximum at the optimal length, تمام? But it's broader. Okay, so that doesn't mean that directly after being beyond the optimal length or shortening the muscle below the optimal length, the active force is going to decrease. Here it is very steep because you have decreased cross-bridging cycles. But here it's very prolonged. Why? Because the muscle is flexible. It doesn't have that much sarcomeres. Okay. Another difference that you can notice here is that the passive tension curve is a lot closer and a lot steeper as well than this here in the skeletal muscle. تمام? It is, why is that? Because when you said passive tension or passive force is when you are stretching the muscle. Like in skeletal muscle, when you're stretching the hamstrings because you are a runner, for example. Okay, and we said the passive tension depends on what? It depends on the connective tissue and the elasticity of the muscle itself. And obviously, the smooth muscles that are in the blood vessels, that are in the GIT, that are in the urinary bladder, and the uterus, any smooth muscle has a lot of connective tissue than the skeletal muscle. Okay, skeletal muscle requires more force and more tension requiring. Okay, it doesn't need that much expansiveness or flexibility. صح? Because why? The skeletal muscle needs to support us. However, the smooth muscle, it needs for accommodation of fluids, the blood, a newborn in the uterus, okay, a baby in the uterus, and etc. So it needs to be very flexible, and that's why it has a high connective tissue around it, and the elasticity of it is even more than the skeletal muscle, and that is why the passive force or the passive tension is higher in the smooth muscle. تمام. Okay, I promise we are almost done. This is again the motor unit recruitment. We said in the skeletal muscle, motor unit is what? A muscle fiber and a neuron. The same thing or a similar thing in the smooth muscle, but in a smooth muscle here, this is one cell, this is one cell, smooth muscle cell, smooth muscle cell, smooth muscle cell. Here, they are connected by this bridge here. These are called gap junctions. Okay, these are basically communicatory proteins or like not channels, routes or tunnels between cells. تمام. And this is one neuron, autonomic neuron, just a neuron, which will simulate these only two cells. Okay, see, it is only bound to these two cells. But because of the gap junctions, the stimulatory wave or the contraction wave can spread to the rest of the muscle. Okay, it spreads to the rest of the muscle through these gap junctions. تمام. And this helps the entire structure to contract as a single unit, which is also efficient. So only one neuron can stimulate two muscle cells, but these two muscle cells will spread the wave of contraction to the other surrounding muscle cells and allow for the contraction of this entire thing as a single unit. And this is called phasic smooth contraction or unitary smooth contraction because it is a single unit. تمام? And this is done by what? Primarily by gap junctions. تمام. Okay, now what if we have one muscle cell stimulated by one individual autonomic neuron? It is called the tonic or multi-unit smooth muscle contraction. Okay, this allows for like specific movements, fine control of everything. So, for example, what the eye muscles, you have the pupils are controlled by smooth muscles. You have pupillary constrictor muscles and pupillary dilator muscles, right? And this needs to be in fine control because it's a reflex, صح? Because that's an autonomic neuron. It's a reflex. When you have flashing lights directly on your pupils directly needs to constrict very fast, and that's why you have a lot of autonomic neurons supplying individual muscle cells, and you have multi-unit smooth muscle contraction, and it is for fine control. تمام? So each muscle is independently innervated by one single neuron. Okay. Alhamdulillah, we are done from explaining. Now let's apply our knowledge in a question. Only 3 questions are here, and we are done, inshallah. Okay, so let's go ahead and answer this question. So the delayed onset and prolonged duration of smooth muscle contraction, as well as the greater force generated by smooth muscle compared with skeletal muscle, are all consequences of which of the following. So take your time, answer it, and then I'm going to show you the answer and explain it. Okay. Okay. So this is the answer. What basically the question is asking you, what is the difference between skeletal muscle and smooth muscle? تمام. So slower cycling rate, smooth muscle, and myosin cross bridges. Why? Because remember the latch contractions when we said that we have a prolonged contraction time for less energy. That is the difference between skeletal muscle and smooth muscle. Okay, greater amount of myosin filaments present in smooth muscle. That is wrong. Actually, we have more myosin filaments in a skeletal muscle because we have more sarcomere or contractile units in skeletal muscles compared to smooth muscles. Okay, higher energy requirement for smooth muscle. That is wrong again because of latch contraction or tonic contraction. It can contract for a prolonged period of time with low energy wastage, okay, or expenditure. Now, physical arrangement of actin and myosin filaments. It is more organized in skeletal muscle because it is more fibrous-like. Okay, slower uptake of calcium ions following contractions. That is not true. Actually, we have more uptake of calcium because we have voltage-gated, ligand-gated, mechanically gated channels and the sarcomere itself in a smooth muscle. Okay? So obviously, depending on the number of channels, we have more uptake or faster uptake of calcium in a smooth muscle than in a skeletal muscle. Okay? Okay, now this is another question. Okay, so which of the following mechanism action is most likely to induce this effect? Okay, let's read the question. An experiment drug is being tested with a potential therapeutic treatment for asthma. The preclinical studies have shown that this drug induces relaxation of cultured porcine tracheal smooth muscle cell pre-contracted with acetylcholine. Which of the following mechanism is the effect? Okay. Okay, basically the question is asking you what relaxes a smooth muscle. تمام? I want you to think about this question very carefully because it is related to the MLCK and the MLCP balance. تمام? So let us show you the question, or sorry, the answer, and let's explain it. Okay, so stimulation of adenylate cyclase. I might have to, to go back to explain this, but let's rule out the choices first. So decreased affinity for troponin C for calcium, that is very wrong because we don't have troponin C also in the smooth muscle. Okay. Or the calcium that enters in the smooth muscle doesn't bind to troponin C. Okay. And decreased plasma membrane potassium permeability. We don't need this here. It's unrelated. And increased plasma concentration for sodium permeability also is not related, so we ruled out these three. And inhibition of ceruloplasmic reticulum calcium ATPase. No, because the muscle is relaxing. Sarcoplasmic reticulum calcium ATPase is basically the circa pump. Okay, if you inhibit it, you will not pump calcium back into the sarcoplasmic reticulum. That means that we have a lot of calcium in the cytosol. This will not cause contraction. This will not cause relaxation at all. It will cause contraction. So this is in case of the question is asking you about contraction. Okay, stimulation of adenylate cyclase. Adenylate cyclase is AC, which will convert cyclic ATP into cyclic AMP. Let's go back here because I want to show you this. Okay, please bear with me 1 second. Yes, here. Okay, remember when I said that phosphorylated MLCK is phosphorylated MLCK has less affinity to the CC complex? This is because there's structural differences and it's not binding properly to it. What causes the phosphorylation of MLCK? If is any pathway which will stimulate, or any substance which will stimulate the cyclic AMP PKA pathway. Okay, so in this question, let's go here. In this question, any stimulation of adenylate cyclase, if you know the pathway of cyclic AMP, you have G protein activating adenylate cyclase, which is an enzyme which converts ATP into cyclic AMP. تمام? If you have cyclic AMP, you will activate the protein kinase A. Protein kinase A will go and activate or phosphorylate the MLCK. Phosphorylated MLCK has less affinity to CC complex, or the calcium calmodulin complex. And if the MLCK will not be activated, it will not cause contraction. That is why this, it's a very indirect question, but that is the explanation of this question. And without any new knowledge to add, we are done from our lecture, and I hope you guys enjoyed it. I hope that I simplified as much as I can this concept. It's a very long lecture, so I advise you to take it at 3 different levels. The skeletal muscle contraction, and then the mechanics, and then the smooth muscle contraction. And thank you guys so much. Please scan the code and give me your feedback for the PAL, for this PAL lecture and the PAL program in general. And thank you so much. And I hope you enjoyed this lecture. Thank you so much, and we can stop now.